OK. Hi everyone. Bonjour tout le monde. Donc, cette session sera en français aujourd'hui. So, today we're going to have an English webinar. Thanks for joining us. And as you can see, the webinar will be recorded and is going to be available on YouTube as always. So, today we have the pleasure to have a presentation by Roseanna Rought. I hope I'm pronouncing your name right. Roseanna is a PhD student in the McGill University Integrated Program in Neuroscience under the supervision of Professor Terence Coder. She is investigating the role of intracellular G protein coupler receptor in the modulation of persistent pain using transgenic mice models, behavioral assays, and molecular imaging techniques. She completed her master thesis in neuroscience at the University of Nottingham with an industrial placement at the AstraZeneca in Sweden, where she focused on cellular assay development. So thank you again, Roseanna, to present to us today. It's really our pleasure to have you here. And I'll stop my screen sharing and I'll just let you share yours.
Yeah, thank you for having me as well. I'm excited to be here and talk about my research with you. Just check. Can you see the full screen now?
Yes.
Okay, great. Shall I get started or do we want to wait a little while?
So just to be respectful of everyone's time, let's start now, and then
okay.
If there's anyone that'd like to join, I'll just admit them in the room.
Perfect. Okay, so yeah, as we said, I'm Roseanna and I'll be presenting the topic of intracellular receptors and their role in pain. So firstly, just to introduce myself a little bit and how I came to study this topic. I completed the integrated master's program in neuroscience at the University of Nottingham, and this combines the bachelor's and master's degree by adding an additional year-long research project. And I completed this project as an industrial placement at AstraZeneca in Sweden, where I worked on a development of an experiment to assess the quality of drug binding to specific targets in cells. And then in August of 2021, I made the move over to Montreal to start my PhD in the lab with Professor Terence Coder. And so a big reason I began my graduate studies in the field of pain was my own experience with chronic pain. So when I was 13 years old, I tripped on a few stairs and tore a couple ligaments in my ankle, and this led to the development of complex regional pain syndrome. So some of you might have heard of this syndrome, but if you're not familiar, it's a chronic pain disorder that can occur following trauma, and it's associated with additional symptoms such as temperature and skin changes in the limb. For example, my ankle was extremely cold to the touch in the later phases of my CRPS. And so this led me to be unable to walk without crutches for about 3 years. And the pain management options available to me were quite limited. So during this experience, I became really fascinated with neuroscience and the molecular mechanisms that lead to the development and maintenance of chronic pain. So this brings me to my project. And today I'd like to talk to you a little bit about the background of chronic pain, the role of intracellular receptors in the mediation of pain, and how we can use animal models to study this. So pain is a vital sensory experience, and it acts as a protective response from actual potential tissue damage. So for example, if we touch a hot pan and we remove our hand really quickly from the dangerous surface, if we have caused some damage, our behavior will change due to the pain to guard the injured area until it heals. And so in this way, acute pain is adaptive and helps prevent or reduce tissue damage. But chronic pain is no longer adaptive or protective. It can be associated with features such as hyperalgesia, which is an increased perception of pain to a normally painful stimulus, and also allodynia, which defines a painful response to a stimulus that does not usually cause any pain. So there are several types of chronic pain, and in my project, I'm investigating 2 categories. The first of which is neuropathic pain, and this occurs following lesion or disease of neurons in the central or peripheral nervous systems. And there's many clinical examples of neuropathic pain. So for example, pain associated with nerve damage due to diabetes, traumatic injuries, or following chemotherapy. So to study the mechanisms of this type of pain, we can use various models in the lab. In my project, I work mostly with mice, and I carry out what is called a spared nerve injury. So in the diagram over here, I don't know if I can the pointer that work there. So here, this shows the sciatic nerve, which runs from your back all the way down your leg. And when the sciatic nerve reaches the leg, it splits up into these three nerves here. And what we do is we tie the common peroneal and the tibial nerve and cut them, but we leave the psoral nerve intact. And this results in a persistent pain model in mice that lasts for several months. And so I'm also investigating inflammatory pain. And there's growing evidence that supports the contribution of neuroinflammation and neuroimmune activation to chronic pain states. So to study the mechanisms underlying pain that result from persistent inflammation, we can again use several models that induce the peripheral inflammatory response. So what I do is inject CFA into the hindpaw of mice, and this is a suspension of heat-killed bacteria in oil, and it triggers an inflammatory response in the mice, leading to thermal and mechanical sensitivity, which can persist for days to weeks following any injection. So before I get into the specifics of my research, I'd like to just give an overview of how we sense and process pain. So a painful stimulus here, such as touching something hot, can activate nociceptors. And these are specialized sensory receptors that detect pain signals. So the pain signal is sent along the primary afferent neurons to the spinal cord dorsal horn. And here is the location where information is transferred from the periphery to the central nervous system. And the signal can then continue along up the ascending projection neurons to the brain. And when the signal reaches the brain, it's processed in various areas. And this leads to both the sensory and emotional aspects of pain perception. But the brain can also send information back to the spinal cord through these ascending neurons. And this helps regulate the output from the spinal cord. So there's many areas in this pain pathway that do have a role in chronic pain, but the spinal cord plays a crucial role in integrating input from the periphery, local interneurons, and descending information from the brain. And so it's currently the focal area of my research. So if we take a bit of a closer look at the spinal cord, it can be looked at as separated in different sections with different roles. So here at the bottom, we have the ventral horn, and this is important for motor processing. Whereas in the dorsal horn, this is more important for sensory information. And the dorsal horn can be split into several sections. So firstly, at the top, we have the superficial lamina, and this has the pain-specific neurons. And then we have the intermediate lamina with touch-specific neurons, and then the deeper layers integrate both the touch and pain-specific information coming from the periphery. And so there are many hypotheses to explain the development and maintenance of chronic pain. One of the most prominent is central sensitization, and this has been really well studied in the spinal cord dorsal horn. So central sensitization is an amplification of the painful inputs as a result of neuroplastic changes in the central nervous system. And this can lead to increased excitability of neurons, which can increase spontaneous pain signals. And so also in normal states, a pain stimulus is usually required to be of a certain intensity in order to initiate a pain signal. However, when central sensitization occurs, this can reduce this threshold. So stimuli of a much lower intensity can trigger the pain. And then finally, central sensitization involves disinhibition. So in the spinal cord, excitatory and inhibitory information is integrated, and this modulates the pain response. But if we lose or reduce this inhibitory information, then more pain signals can be transmitted. So a key area of interest to explain the mechanisms underlying the central sensitization is the alteration of glutamate signaling. So glutamate is one of the major excitatory neurotransmitters in the central nervous system, and it's important for the normal transmission of pain from the periphery to the central nervous system. But it also does have a role in central sensitization. So glutamate can activate several types of receptors, and these fall into two categories. So firstly, we have the ionotropic receptors, and this includes NMDA and AMPA, and they're channels which open and allow positively charged ions into the cell when they're activated. And in the clinic, we have a lot of antagonist drugs that have been developed that inhibit these receptors by blocking the channel from opening, and they are effective in reducing pain, but they're also associated with quite severe side effects, such as hallucinations and impacts on motor control. So this really does limit their use in the clinic, because obviously people don't want to be having these side effects while they're trying to control their pain. Therefore, my lab, among others, have shifted their attention to the other category of glutamate receptors, which is the metabotropic receptors. And my project focuses specifically on mGluR5. So mGluR5 is a G protein coupled receptor. So that means it's associated with these G proteins that you can see here. And when glutamate binds up here to the receptor, it causes a shape change in a receptor and it becomes activated. And this activation results in the release of these G proteins. So you can see here that they're no longer associated with the receptor, and they can go on in the cell to generate secondary messages that create a signaling pathway in the cell. So it's traditionally been considered within the field that only GPCRs on the cell surface are functional. However, there's now growing evidence that a large proportion of these receptors are present and functional at intracellular membranes. And these receptors can be found at different locations within the cell. So we have them at the nucleus, the mitochondria, and the endoplasmic reticulum. And these intracellular receptors often result in different signaling pathways compared to the cell surface receptor. And this is known as location bias. And so the existence of these intracellular receptors that are capable of different signaling, it represents an opportunity to develop selective therapeutics with the potential to maximize their effectiveness and reduce off-target side effects. So to focus in on mGluR5, on the right here, you can see an image I produced from the fluorescent staining of mGluR5. And this brighter band at the top indicates the presence of a receptor in the superficial lamina of the spinal cord dorsal horn, which, as I mentioned earlier, contains pain-specific neurons. mGluR5 has an established role in the modulation of pain transmission, and antagonists which inhibit the receptor are effective at reducing pain. But it has recently been shown that mGluR5 is also present on intracellular membranes. So in fact, at least 80% of this receptor is found on the intracellular membranes, including the nucleus and the endoplasmic reticulum. And this receptor can be activated by glutamate, and it is functional. But what is interesting for my project is that these mGluR5 levels are increased at the nucleus in the models of both neuropathic and inflammatory pain I mentioned at the start of the talk. So as I mentioned earlier, intracellular GPCRs can show a location bias in their downstream signaling that they initiate when they're activated, and those are different to those associated with the cell surface receptor. So not to worry too much about what these different proteins are, but just here we can see that these molecules are activated by the cell surface mGluR5, and these ones over here are the ones that are associated only with the activation of the intracellular receptor. And importantly, these targets of the intracellular receptor are associated with pain transmission. So we have a protein called ARC that is involved in synaptic plasticity, and this can affect the transmission of pain signals. We have FOS, which can sensitize neurons to pain signals, and ERK, which through various mechanisms can increase the excitability of the spinal cord dorsal horn. So this indicates to us that when the mGluR5 is located intracellularly, it may be having a different effect in pain compared to when it is localized at the surface. So far we have covered that mGluR5 is upregulated at the nucleus in animal models of persistent pain. And this receptor, when it is at the nucleus, is able to result in different pain-relevant cellular effects compared to the cell surface receptor. So therefore, a previous PhD student in my lab looks at the impact of upregulated nuclear mGluR5 on pain behavior and signaling in rat models of persistent pain. And she investigated this by exploiting the permeability of different antagonists of mGluR5. So we can see in this image here that we have permeable antagonists that can block this cell surface receptor, but they can also get into the cell and block these receptors that are on the inside of the cell. And it was found that these permeable antagonists were really effective at reducing pain behavior in these animals with persistent pain. And also they were able to reduce the levels of both the targets of the cell surface receptor and the intracellular receptor that I mentioned on the previous slide. However, when we use impermeable antagonists, which can only target these cell surface receptors and they can't get into the cell to target the receptors at the intracellular membranes, they were a lot less effective at reducing both the pain behavior and the levels of intracellular signaling targets. So this demonstrates that inhibition of intracellular mGluR5, but not cell surface mGluR5 alone, significantly reduces pain behavior and signaling in persistent pain models. But there are some limitations to the manipulation of receptors with antagonists. Several of the drugs used in these studies were not highly selective. So this means that they may also be binding to and affecting the function of other receptors. And therefore, we can't be sure that the results are solely due to action at mGluR5. This is important to know, as if in the future drugs are developed to target mGluR5, we need to be sure that they will have the behavioral effects we are looking for, as well as not causing unexpected side effects. Another limitation of the previous work on the role of mGluR5 in pain is that the majority of it has been done in male animals only. Clinically, females have a higher prevalence of chronic pain, but there's been a historic lack of research in the field into the mechanisms underlying the differences in female pain. mGluR5 is known to interact with the female sex hormone estrogen in several other biological processes, such as addiction, so it's possible there may be some sex differences in regards to pain, and it's important for us to look into this. So therefore, my project aims to assess the pain behavior and downstream signaling of mice of both sexes with mGluR5 genetically restricted to the intracellular membranes. And for the rest of this talk, I'll talk about these mice as IM mice. So we've hypothesized that the IM mice will display pain hypersensitivity and increased mGluR5 associated spinal cord dorsal horn signaling following the neuropathic or inflammatory injuries. So just to give you a bit of an idea about how these mice were generated, they were made by our collaborators, and they use a technique called CRISPR, which is a method to edit genes. So to do this, they used another receptor called the lamin B receptor. And this receptor is found generally in the nucleus of cells in normal mice. And it has a sequence in its genetic code that helps the cell target it to the nuclear and endoplasmic reticulum membranes. So using this gene editing technique, they took the sequence from the lamin B receptor and attached it as a tag to the mGluR5 receptor. And now this means that all the mGluR5 receptor in the cell is restricted to the intracellular membranes and not on the cell surface. So this means that we can study mice that have no cell surface mGluR5 receptor to establish how important the intracellular receptor is in persistent pain without the use of any drugs that may be having effects on other receptors. So these mice that have been developed are normal in terms of lifespan and most general behavior. But if we look at this image here, which was produced using an electron microscope by our collaborators, so all these little black dots here are representing the receptor inside a neuron. And we can see here, it's labeled the plasma membrane. This just means the cell surface membrane. There's no little black dots. There's no receptors present here. Whereas when we move to the nucleus and endoplasmic reticulum, and it's just zoomed in here, there's a lot of black dots present, which shows that the receptor is only localized inside the cell. So now I'll just talk you through the experiments I've conducted so far in my studies and talk a little bit about the results and what we're seeing. So to assess the pain behaviors of the IM mice, I first conducted the formalin test. And this test is frequently used to study continuous pain generated by tissue injury in rodents. So we inject formalin into the hindpaw of the mice, and this results in a biphasic response of licking and flinching behavior. This behavior tends to last about an hour, and it's broken up into two phases. This early phase, which lasts 5 to 10 minutes, is thought to be caused by activity in a subset of the primary afferent neurons that go from the periphery to the spinal cord dorsal horn. And then the late phase begins about 15 to 20 minutes after the injection, and it appears to be dependent on both inflammatory processes and functional changes in the spinal cord dorsal horn. And then in separate groups of mice, I injected CFA, which is the inflammatory model I mentioned earlier, and I assessed their pain responses in various behavioral experiments over the following 4 months. So firstly, I looked at the von Frey assay, and this is an assay we use to look at mechanical induced pain. And it involves administering filaments of various forces. And these filaments just look like the bristles on the hairbrush that we apply to the paw of the mice. And when we apply them and it's painful, they withdraw their paw. And so we use what is called an up-down method, and this helps us calculate the 50% withdrawal threshold. And what that means is that the stimulus strength required to result in a withdrawal response in 50% of trials. And this just gives us an idea of the level of mechanical allodynia that these mice are experiencing, as often when they're injured, they respond to filaments that they would not normally respond to. So they're getting a pain response at a much lower intensity. And then to assess hypersensitivity to thermal stimuli, I use what's called the Hargreaves apparatus. And this is very similar to von Frey, but instead of a mechanical filament, we use heat. So we just apply a heat stimulus to the surface of the hindpaw and measure the time taken for the mice to withdraw their paw. So when the mice is in more pain, they withdraw their paw from the heat a lot quicker. And then finally, I used the rotor rod test. And this just involves placing mice on a rotating rod and measuring the time it takes to fall. And this isn't a measure of pain, but it's just used as a control to make sure the motor performance of the mice is not impacted by the different genotype. And this could, because this could impact their performance in the other pain experiments. So I just wanted to finish off with a little summary of the data I've gathered so far and what my next steps are in this project. So I found in the formalin test that the IM mice spend more time in pain. And interestingly, the female IM mice have a longer duration of pain than the males. And this data was supported by findings in the CFA model. So in the normal mice, they recover from the mechanical allodynia caused by the CFA in about 2 to 4 weeks. But the IM mice took 4 months for their responses to return to baseline levels. So this shows that restricting the mGluR5 to the intracellular membranes is having quite a big effect on the duration of the pain response. But there were no differences between the normal and the IM mice in terms of the thermal sensitivity. But this can also be the case in clinical conditions where thermal sensitivity is not always a feature of chronic pain. And then interestingly, when I injected CFA into the left paw of the mice, I also saw increased mechanical pain responses in the right paw of the mice for 2 months following injection. So this implicates mGluR5 in mirror pain. And mirror pain is a process that is seen in some clinical chronic pain conditions where you injure one leg, for example, and then you get pain in the other leg or an arm. But the reasons why this happens are not well understood. So I'll definitely be investigating this further to see what the role of mGluR5 is here. So just to conclude, the data I've gathered so far supports the importance of intracellular localization of mGluR5 in the persistent modulation of pain and mechanical allodynia. And so my next steps are just to repeat these behavioral experiments in a neuropathic model of persistent pain to establish the role of mGluR5 across various pain types. And I'll also investigate the injury-induced downstream signaling of mGluR5 in the IM mice. So thank you very much for listening, and I'm happy to take any questions that you have.